This is the muscle. It has a muscle belly, fascicle, fiber, myofibril, and sarcomere, all made of filaments. Every sarcomere contains overlapping actin and myosin filaments. In order for the muscle to contract these filaments, they must slide past each other, resulting in the shortening of muscle fiber. This is known as the sliding filament theory. Muscle contractions start when someone tells it to move.
It starts with an action potential. The motor neuron responds to the brain's order and sends down the electrical signal. The action potential reaches the synaptic terminal, or the axon terminal, which is the end of the axon and where the neuron and the muscle fiber meet. Acetylcholine, which is a neurotransmitter, is released into the synaptic cleft of the neuromuscular junction. It then binds to the motor end plate, which changes the permeability of the membrane to sodium ion.
Sodium ions spread the action potential across the surface of the sarcolemma and down the transverse tubules to the terminal cisternae, a part of the sarcoplasmic reticulum, or the SR, which releases calcium ions.
These calcium ions go a long way. Calcium is essential for muscle construction. It binds to the protein troponin, which hold the protein tropomyosin in place. Tropomyosin covers the active sites of actin. When calcium binds to troponin, it changes position, moves the tropomyosin, and uncovers the active site of actin, which allows the myosin head to lock on to actin, forming a cross bridge. Calcium enables the exposure of the cross bridge sites, which is essential to making the contraction possible.
The head of each myosin unit is bound to an ADP molecule remaining from the previous muscle contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. As the units move, the ADP molecules bound to their heads. The gliding motion is stopped when ATP molecules bind to the myosin heads, destroying the bonds between myosin and actin. ATP molecules break down into ADP plus P, and the energy released by this reaction is stored in the myosin head for the next movement. ATP then moves the myosin in a powerful stroke, bends the thin actin filament, and pulls them on top of the thick filament. This is the contraction.
The sarcomere has A bands, which contain the thick myosin filaments, and I bands, the region between two consecutive A bands. The way bands react, the I bands disappear and the A bands move closer together, imply that thin filaments slide towards the center of the sarcomere, closer to the thick filaments, which is how a sarcomere will contract. What about its relaxation? Well, to get the myosin head to release, a second ATP is needed. Calcium is then taken back into the SR, and tropomyosin returns to its place and covers the active site, allowing the muscle to relax.